extra glucose, and it itself, without seeing insulin, can do this. So people that have type 1 diabetes are still able to get extra glucose in their muscle, even if there's no insulin left. And for people with type 2 diabetes, because this is not dependent on insulin, there's no issue with insulin resistance, right? So it helps both types of diabetes to exercise because the muscles have developed to do this. And that's why we are always like hammering on the fact that people with diabetes need to exercise regularly. Just wanted to make sure that that is out there, and I made a note on the slide so that next year I shall not forget. Thank you. Somebody came up to me afterwards and asked me a question about this. All right. Are there questions about this or yesterday's lecture in general? No? Okay.
She wants this on Friday.
So then, let's move on to today's lecture. So today is the last lecture that is part of next week's midterm. So however far we get, that's how far we'll get. I have an endpoint in mind and I hope we make it there. If we don't, then whatever comes after will obviously not be part of your midterm. It's kind of awkward that this is a set of 2 lectures that we might be chopping in half, or we will be chopping in half. Yes?
So like the
Maybe,
three, I think the midterm will be 20, 32, is it?
Oh, did I write that? I don't think so. Let me see. So God, I tried to chop it into sets of 10.
I just want a piece of this right now.
No, that's not what I meant.
Oh,
I meant like copy your picture.
but then I need this one.
Go to front.
It does say that here.
Go to front.
Is it for skipping something? I think because like, one pop out.
Oh, awesome, I didn't even know.
This is my PDF that I probably also used to upload. This is not correct. I tried to have chunks of 10, and this is what we did in a previous duration. We can even talk about this right now. So I was going to let it run from 21 to 30. So that makes today's content the last. And you have some time to digest it before we actually hit the exam. And then these 4 lectures, and whatever comes after there, are just going to be part of the final. I'll make an announcement to make sure that everybody sees that. Can you remind me, Alex?
Monday?
So this should be 21 to 30.
I gotta go home by like 5, the latest.
Alright, so today we'll talk about epigenetics, and epi kind of means on top. So this is things that layer on top of genetics to determine how genes are expressed. And this is a fairly new field. We didn't really know about this, I don't know, 20 years ago maybe. The central dogma is that you have DNA in your nucleus, it's transcribed into RNA, and then that's made into protein, and that is what has the effect. And what we've come to realize is that there are ways that we change the way that the code is read that have nothing to do with the sequence of the protein, with the sequence of the regulatory sites, but it has everything to do with changes to the whole thing of chromatin. So either the DNA itself or the proteins that it's packed up with that change the way that the genes are used. And so that's what this lecture is going to be talking about. I think these are fairly straightforward learning objectives. Let's see. I think I'll try to get us today to here so that we know all about the agouti gene and what happens, and that we'll save those for tomorrow. So this is another one of those integrations, and this slide might look familiar from the early lectures where we've tried to make this class so that we talk about things that, yes, are often very much at the cellular level, but now you get to see how it affects the whole organism, right? And so these pictures are here. What do animals need to do to be successful? We've learned about a lot of cellular mechanisms, and now we're going to try and get them back up to the whole organism. So we should not be eaten by other organisms. We should slaughter ourselves, feed ourselves, and also reproduce. If you don't reproduce, you're not successful in the evolutionary sense of the word. In order to be successful, you have to have a good number of surviving offspring, and then the way to get there is by to have your genes match what the environment asks of you. And this is not always straightforward, because if you have a certain gene set that gives you a nice thick furry coat, that's great if you're living in Alaska, but maybe not so handy if you're living down closer to Mexico, where it can be really, really warm. So there you'll need different mechanisms. So the same phenotype that increases fitness in one environment is not always the best for another. The phenotype, as we know, comes from our genotype, but we do see these things where even though some organisms might have the same genotype, they still don't have the same phenotype. There's some plasticity, some differences possible starting off with the same genotype. So here we have an example of some identical twins. So they would have started off as one fertilized egg, started undergoing mitosis, and then it split into two embryos. So they have the exact same genotype, but yet in this case you can tell, and this is not due to a nice high hair dye from the package, but this is just what their hair looks like. Definitely differences in color there. Another example here, this dog has one blue eye and one sort of more green-brown eye. And this is something we see in humans too. There's some famous examples. If you look at close-ups of these women's pictures, you see that their eyes are different colors. And here's a very interesting example. Here is the first cloned cat. Her name was Cece, the copycat, which I thought was kind of cute. And this is her mom, her genetically identical biological mother. And see that they have differences in what their coat looks like. And that's because the color of the hair is determined by somewhat random process occurring as the embryo develops. So the same set of genes was used slightly differently. For the hair color, the cells that make this darker hair colors, they actually start off at the back of the embryo, and then they have to migrate down. So if they don't make it all the way down, the hairs at the bottom of the belly, they stay white. So if they start migrating a little bit later in one embryo versus the other, then they have a larger white belly, right? So yeah, cool stuff. And so some of this is then due to epigenetics. Epi means above or on top of. And so these are changes that do not involve changes in base pairs. There's no change in the order of the A's, C's, T's, and G's, but it's other changes that are made to the DNA or to the proteins that it's packaged in that are going to drive or inhibit expression of certain genes. And so we can see this process occurring just in normal development. As we talk about stem cells too, cells that have the opportunity to become a lot of cells, as they differentiate and further restrict themselves, you'll see that some genes will get silenced through this mechanism of epigenetic modification. So it's a normal process that happens a lot, and today we'll learn a little bit more about it. We won't go into great level of detail, but we'll have some interesting examples. So how can we pass information from parents to offspring? Obviously, a lot of that goes straight from the DNA sequence, but we can also chemically change the chromatin, and that can also be inherited. So we have three mechanisms. We'll mostly talk about these top two, and we won't really focus on this last one at all. But here's the three. So we can change the way the DNA is packaged. As we saw here in this picture, here's our double helix winding around and around. And you can see here that the DNA is not just kind of winding around itself, but it's also wrapped up around these proteins. If you were to take the DNA that is in one of your cells and just roll it out all the way, it's longer than you, for most of us, I would say, because you'll end up with about 2 meters of DNA. And instead of having this long, long string of unwound DNA, our cells package it up into a unit that we can barely see, right? We need a microscope to see. And they do this by winding it around different proteins, histones. And if you wind them up really tightly, it becomes harder for proteins to come in and drive transcription. So if the DNA is really tightly wound up, we call that heterochromatin, and it's not as easily accessible. So if you change the way that the DNA is packaged, if it's more tight versus more loose, you can already see that it's easier or not as easy for other proteins to come in. And we do this in large part through changes, modifications to the histones, those proteins that the DNA wraps around. The other way you can change the expression of genes is by DNA methylation. So this here is done to proteins, the histones that the DNA wraps around. You can also directly put a methyl group on DNA. This will inhibit proteins from coming in and generally results in an inhibition of transcription. This final one, non-coding RNAs. So the central dogma that I just mentioned has each message come in and code for a protein, and now we have a protein that's functional and can do stuff. But we also have these messages that do not code for a protein, and they can actually interfere with the expression of other genes. So if for the other genes a message does get made, these non-coding RNAs can sometimes come in, bind, and then thereby initiate this process where the message is destroyed. So even though the transcriptional machinery is active, we don't see a protein made from these messages because those interfering non-coding RNAs are preventing the process of translation by breaking down the message before it gets translated. Right, but like I said, we'll mostly concern ourselves with these top two. Just know that the third one is definitely something that's going on. So, how is DNA packaged? Like I said, it wraps itself around these nucleosomes. So we get these loop-to-loop, I think it's about 1.75 windings of DNA around each histone. Then there's a bare piece of DNA, and then the next histone loops. And we saw this actually in our apoptosis lectures, right? If the cells are going to go undergo apoptosis, these DNA cutting enzymes come in, and they are able to reach this area, but not so well this wound up area of the DNA. So they'll chop it here, and then we get these regular sized pieces, 180 base pairs or so. This is the case if the DNA is accessible, so if it is wound up but somewhat loosely. We call this U chromatin. U means real, so this is the real stuff, the stuff that's actually used for transcription. Proteins can access the DNA because it's more available, whereas if you package it up even more tightly, it becomes heterochromatin, and then that's less accessible, so there'll be less genes expressed from those areas. Let's see. So if this wound up area happens to be the area that can normally start transcription, then you can already see that that is not going to be as easily accessible. And there are also sequences that attract proteins that can actually help make the DNA locally less or more wound up, and thereby they can change the way that the DNA is used. So this is our protein modification that affects how genes are being expressed. So those histone proteins can be acetylated, and this favors euchromatin. And you can imagine this is what a piece of this histone normally looks like. Here we have a lysine amino acid, and we have this positively charged end group there. A DNA is negatively charged, so you can imagine that if the histones all have this positively charged lysine sitting there, and the DNA is negatively charged, that they like to interact, that they like to bind, and it gets more tightly wound up. If instead you acetylate this lysine, put an acyl group on there, so the positive charge disappears, it becomes more neutral. There's less of an interaction between the DNA and the histones. So now that means that there's going to be a less tight, wound up situation, more open, and the DNA is accessible for transcription. So that's why we mean it favors euchromatin. It favors that more loose orientation of the proteins and DNA. There's enzymes that actively acetylate the lysines, and then we also have these HDACs that actively deacetylate. I think Alex, you were telling me one time that for the post-traumatic stress disorder, people were now trying to use those HDACs, or was this an older mechanism?
No, they are.
They were? So yeah, so they're now trying to use activators of these enzymes to try and remove some of the imprinting that could have occurred in post-traumatic stress disorders, to try and relieve some of those symptoms by changing the way that the genes have been allowed to be more expressed. So changes of histones, changing the way that the DNA and the proteins interact, opening everything up, expression increases. Yes?
I have a question for the previous slide.
Yeah.
I was confused if this is a lysine amino acid.
That's part of the histone protein.
Oh, okay.
So the histones, they are sort of these disc-like proteins that the DNA wraps around, but they also have the ends of the protein that stick out and are very mobile. And that's where these modifications often are put on those histone tails, and they are free to stick out and interfere or help with packaging of the rest of the DNA. So DNA methylation preferentially occurs on the cytosine basis of DNA, and this one favors the DNA to not be used. So if we look at the DNA molecule, we can add the site group. Now you can imagine it looks different, right? So if normally some transcription factor would come in and it fits nicely here on this base pair, now it has this side group there and it won't be able to bind there as easily. So this is preventing that thing from binding, and it also serves as a signal to recruit silencing proteins. So they will come in and maybe once they sit down on the methylation, they start changing the histones, which then become more tightly packed. So, for example, those deacetylases are going to be recruited onto these methyl groups. So now we have the start, and this is going to make sure that the DNA gets more tightly packaged because they are going to be removing the acetylation, and now we're back to a positively charged side chain for this histone. And so these things don't occur in isolation. We'll have acetyl groups going on, we'll have methyl groups going on, and it really is the overall total of these things together, which one is winning, that's going to drive whether or not you see transcription occur from this area of DNA. And so we can have methylation occur, which recruits those deacetylases, which will make the DNA more tightly packed. So now we have fewer methyl groups and fewer acetyl groups, and the DNA becomes much more compact. So they really are working together to drive these changes. If I were to ask you guys something on the midterm, I would give you not just the name of whatever enzyme, but also what it does. So I would ask you guys to know what the effect would be of this acetylation or deacetylation. So don't worry about the name so much. So not all cytosines in DNA are methylated, only a small percentage. But there are regions in our DNA that are very enriched for those CG sequences in which the cytosine, the C, is then methylated, and we call these CPG islands. So those are often located close to the promoters. Promoters are sequences that are going to be driving the transcription of certain genes. And so many of those areas are methylated to silence the genes that are located in proximity. Right, and so we draw this out, like here's a chunk of your chromosome, here is the coding area of the gene, and then here we have a CPG island that is either, if we use the white balls, there's no methyl groups, but if there's these black balloons sitting on top, it's methylated. And so you already know methylation will inhibit the transcription, so if you see much of those black balloon sitting on top, that indicates that this gene is going to be relatively silent. Not absolutely quiet. Usually in biology, nothing is black and white, right? And oftentimes we'll see a couple of methyl groups here and a couple of unmethylated cytosines there as well. But this is the extreme case. So here is an example then of histone 3, H3 in short, with that tail that's exposed, and you see that there are many lysines and serines that can be modified. We talked about methylation and acetylation, but we can also see phosphorylation. So kinases can come in and phosphorylate different amino acids. Serine, threonine, and tyrosine are most often those that become phosphorylated. So this is going to add a negatively charged site group, right, to those DNA, to this histone, and therefore have an effect on how this DNA is used. So if we know that phosphates are a negatively charged site group, can we imagine what the effect would be based on what we've seen in this lecture and also in other lectures involving phosphorylation?
age we just observed now.
I'm guessing C. I don't know. What does Karen think it is?
Alright, so this is a little bit of an open-ended question, and you guys show that really nicely. As I was thinking about this without further looking up what the real story would be, to me, both of these would seem to make sense, right? We hung up this whole story about the charges on the histones being important for how tightly the DNA gets packed. So removing a positive charge by acetylating it would make it more tightly, or less tightly packed. So I imagine adding a negative charge would even make that more strong. And we also have seen in earlier lectures that if something becomes phosphorylated, this is often now an attractive site for other proteins to come in and bind. So we can imagine that that's the case too. And then I put the other in there because as I was reading, it actually turns out that we don't really know all that. How do I put this? Can everybody just put C so we have a uniform answer? That makes it easier for me after. If you have D, I'll ask you to offer an explanation. Where is it? Here. Alright. So in fact, we don't really understand all of those signals yet. So while it makes sense that phosphorylation maybe relaxes the chromatin, what actually seems to be happening is that phosphorylation is involved in the condensation of the DNA as cells undergo apoptosis. And we also see sometimes if some serine gets phosphorylated and then locally a lysine gets acetylated, that combined signal actually helps drive transcription. So some of it seems to be the way that we think, and other things go through unknown mechanisms. But there's much to be learned still. Alright, so we all start off with some level of epigenetic change, and in the case of monozygotic twins, they actually start off with the exact same DNA. And here they did this experiment where they were combining, so these are human twins, different human twins, sampled at different ages. And you can see here, so these are two samples from twin A, two samples from twin B, and then the same in this one, except these are 50 years old and those are 3 years old. And what they've done is they've isolated the DNA from some tissues of these twins, and then they've digested it with an enzyme that is sensitive to methylation. So if an area is methylated, the enzyme can't chop it, but if it's free and the right sequence is there, it can. And so if you then do a reaction where you amplify the DNA to see what size of DNA is represented in your sample, you can see that early on, the methylation really isn't different between these children, but as twins age, you get these different sized bands in their samples. So what this means is that the methylation on their DNA must have been different. Otherwise, this enzyme would have generated the same pattern. And you can quantify the number of different bands that you see in young or old twins, and you can see a huge increase, right, in the number of differences. So this means that as these twins are aging, they experience different things. Maybe one goes to college in one place, the other goes to college in another place, the local food is different, who knows, right? They experience different things. And this you can now see in the epigenetic modifications of their DNA. So that's actually pretty cool stuff. Don't worry about the technique. I'm not going to ask you about that, but the idea is that epigenetic modifications are not stationary. That's what you should take away from this slide, right? They change with time and with what you do. So one example of what we do is the epigenetic differences that start to pop up by the things that you eat. These are examples of the mice that we'll go into more detail on. There's also this example from my home country, the Hunger Winter of 1944. My country was occupied by the Germans, and the Germans tried to conquer us, and also by just drying up all sources of food. So we were actually eating our famous tulip bulbs instead of growing them for their pretty flowers. And this block of the food supply resulted in a huge drop in how much calories were consumed for a large population. And children that were born around this time, so like my dad, he's born early 1945, so he's definitely a Hunger Winter baby. They're smaller, they had many diseases, anemia, diabetes, depression, and you can imagine that this is due to the things that they experienced in the uterus, maybe, right? They didn't have all the nutrients that are necessary for normal development. But interestingly, women going through this hunger winter that maybe had children many times later, their children showed the same sort of effects as the children that were born right at the end of the war. And so this has to do with somehow the effect of this huge reduction in calories, modifying not only changes in the chromatin of these moms, but also in their eggs. And this you can still see in their offspring. So some of those modifications are maintained. So something that's more easy to study is this example in mice. These mice differ only in the expression of one gene. Isn't that amazing? You can not only see that this one's more yellow and this one's more brown, but you can also see that this one is a lot bigger and fatter than the other one. All of that because of one gene. And so I wanted to spend a few minutes drawing out the picture that we'll get to on the next slide, because I feel like it's very confusing. It has a couple things that come together, and if we just build it up step by step, it will be much easier to follow that. So the gene involved is called agouti, and we'll see that we have a healthy copy that we'll call A, the agouti gene. And so capital A is the wild type gene for this thing. Anybody see that? Yeah. And then we're using a small letter. It's also the agouti gene, but this is a mutant that's not functional. This is fairly typical of how we do this, right? The capital is the wild type, and then the recessive allele is a small letter. If we look at the gene structure that they'll give you in the pictures, we see some promoter sequence that drives the expression of this agouti gene. And the agouti protein that comes off is a paracrine signaling molecule. And its normal function is to help with the coat color. So it tells melanocytes, those are cells that put pigment in your hair. Tells melanocytes to make a certain pigment, and in this case, a yellow pigment. Right, and so here we have regulatory sequences, the promoter, and so what normally happens is as a hair is growing, it's coming out of the cell that's making it. So as the hair grows, the agouti gene is initially off, then on, and then off again. So if you were to draw what that does for the hair, here I'm going to put my little hair. If the agouti gene is on and this yellow pigment isn't made, the tip of the hair comes out black. Then the agouti gene is turned on and we get this yellow piece of the hair, and then it's turned off again and the hair grows black. So if you look at this on the whole mouse, the mouse kind of looks brown, but if you look at each individual hair, it's multicolored in different layers because of this turning on and off of the gene. So if it's on, we get the yellow pigment. And if it's off, we get the black pigment instead. Right, so that's what we see there and the black line. So this is what normally happens. The mutant gene, let's put some space in between here. The small letter A also has the normal regulatory sequences, so it would be turned on and off, but the gene is broken, so the signal isn't made. So what would this hair look like if there's never any yellow pigment? It would be all black, right? So these mice are all black. Very simple. And now we get to the actual part that this lecture is about, but now I think we can kind of understand it better. The functional gene, so the capital A, has this isoform that they labeled GY, and we will not concern ourselves with what that all means. But what happened there is in the regulatory sequence, a piece of DNA inserted itself. So we have the functional agouti gene, but here they call this the IAP, which is, can you still see this? Yes. A transposable element. So viruses sometimes come in, they insert their own DNA in our DNA, and then they try to tell our cells to make their things. And they also make use of those transposable elements. They just insert themselves into our DNA and then try to drive expression. So in this case, one of those element style things is inserted, and what it does, it drives expression all the time. And not just all the time, but also all over the place. Right? So, we call that ectopic, out of place. So now, if I grow a hair and I have this isoform of the agouti gene, what's my hair going to look like? It's going to be all yellow, right? Because we have the agouti on all the time. There's never a chance for the black pigment to make the hair black. So that explains the difference in coat color for this gene. The hairs are all yellow. But because it's all the time and all over the place as well, this is apparently a very strong driver of expression in different tissues. We don't just see effects in hair. There's also effects that are mediated by our brain. So we see diabetes, obesity, some forms of cancer. So this thing is just expressed all over the place. Now, because viruses do this to us more often, our bodies have mechanisms in place to try and silence these transposable elements. So our cells oppose the IAP. And we do this, and we get this, now we get back to our lecture. By what do we want to the DNA? How can we silence DNA by adding a group to the DNA? Was it the acetylation or the methylation? Methylation. And now we get back to normal regulation. And the air is multicolored. Right, so cells that have this IAP inserted, they try to oppose it, but you can imagine that not every cell or not every animal has methylated this area to the same extent. So now we get to this gliding scale of different coat colors. Okay, we'll change this back. So here we have some mice. They all have one mutated copy that is not going to give us any agouti ever. So we really only have to concern ourselves with the other copy of the agouti gene, and that in this case they have that AY copy which has that insertion name. So if you don't methylate this, the IAP is very active and is driving the expression of the yellow agouti gene in every cell pretty much where it can. So in way too many places, whereas if you've methylated this area, now you get normal developmental expression of the agouti gene in the right spot. And so now we have that on-off timing, and the mouse's coat is going to look more brown. So if you line the mice up, these are all of this same genotype. The only difference is in how well methylated this area is. So very unmethylated, you get a very yellow mouse that's also obese and has all these other issues. Whereas if you methylate it strongly, you get to this more brown mouse. And you can also see that it's not the same over the entire coat, right? There's areas here that are darker. So even within one mouse, you see changes. And now the cool thing is, we can change by feeding these mice different things, how well this area is methylated. Yes? It's a transposable element, so I think those may have come originally from a virus, but now it's no longer the full virus, but it's still a piece of DNA that can jump around in your genome. And so for this allele of the agouti gene, it has inserted itself there and it's driving the expression of the agouti gene. Yes?
Does that mean that it can start off in one place and go to another, or that it could just be found in different places in different
So if it's still able to jump, it could jump out of there, and then if that happens in a germ cell, that this mouse's offspring would have sort of more of the wild type allele for this thing. I think we have quite a few of these jumping around in our genomes, and they're not stable. So if, where are we, a couple minutes. If we take a black mom, so she would have the non-functional gene, right? Never making agouti. And we cross her to a dad that has one non-functional copy and then one of this special isoform of the agouti allele. The offspring that has gotten from their dad this special allele is going to look a certain way. There's differences, but if we supplement the mom with folic acid, vitamin B, choline, and betaine, it shifts the coat color towards brown. So what does that mean for what's happening to the gene? Does it become more or less methylated? More. It becomes more methylated. These things are helping methylation. They're methylation donors in that sense. So if you breed these two together, you could expect all of them to look pretty yellow, but if you give the mom these methyl donors, the offspring will become more brown because there's more methylation in this area. If you don't feed the mom, then there's still a range, but more of the offspring will be this yellow because the agouti gene is allowed to be expressed in more places, right? So these kids look a certain way because of what their mother was eating, right? So what you are eating right now might affect what your kids look like. And wait, that's not all, because what your grandma was eating might affect what you are right now looking like and how you are responding to stress, right? And this is because here is the mom that we had as our black mom in our experiment before. If we say supplement her, and the baby that's growing inside her tends to be more brown, whereas the babies from an unsupplemented mother tend to be more yellow. These girl babies are also making their eggs already, right, in the womb. So they are also exposed to this supplement, and so their eggs are also tending to give rise to more brown offspring. And this is true even if when they are moms, you supplement them. You can still see the difference in range. Supplemented to supplemented is more brown versus unsupplemented to supplemented is more yellow. Right, so it matters what you eat, what your mom ate, what your grandma ate, kind of crazy stuff. And that also brings us back to those people in the Dutch famine cohort, right? Girls that were developing back then would also have seen maybe some of these changes laid down in their eggs, and therefore we can expect to see these effects continue on in multiple generations. All right, that's as far as I was looking at today.